ILRI is producing seeds to make them available for partners and farmers to use to grow forages to mitigate feed shortages. Every year, over 2,000 samples of seeds are distributed worldwide for forage research and development.
We are testing different Desmodium species, which we initially got from ILRI, a germplasm bank. We are trying to see their disease suppression abilities. There are about more than 10,000 farmers who are now doing the push-pull technology, and farmers are doubling the yield, producing the napier grass and Desmodium for the cattle.
I'm a widow. I have a family of 5 and 3 cows. I started growing Desmodium because it chases away the stem borer infection from the maize. At the same time, it is of much use to my animals. Before, I used to waste a lot of money buying napier for my cows and maize for my family.
The ILRI germplasm, or propagatable plant material, is also used for gene discovery. This is the time-consuming scientific process necessary to assess and isolate the important genes inside plants. The science behind the characterization of these fodder and forage crops is labor-intensive. So much of the ILRI collection still awaits gene discovery. But the need for plants resistant to drought, disease, flooding, or temperature extremes is growing, and the stored seeds can supply many answers.
Fodder crops are effectively a buffer to deal with feed deficits in harsh times in smallholder systems. We still don't know how climate will really affect the different environments. Conservation of genetic diversity in crops is extremely important.
Both developed and developing countries are vulnerable to disasters which threaten the food needs of their livestock. The Genebank's library of possible solutions could be invaluable to their futures.
The great resource in the Genebank in Addis could be linked to the new laboratory facilities being built in Nairobi. This offers the opportunity to use modern molecular techniques to better characterize the accessions in the Genebank and look for useful traits. For example, many of the accessions are grasses, and if one can find grasses which have this trait for drought tolerance, that could be used even in breeding with not only fodder crops, but even with crops such as maize and sorghum.
Rich and poor will all continue to need forage crops. But, faced with today's large-scale, fast-moving environmental challenges, they also need networks of dedicated gene banks and their scientists.
In Brazil, we have different biomes that imply that you must have different kinds of forages well adapted to each of them. That's why having a gene bank such as ILRI's is so important, not only for a country like Brazil, but for the whole world, in order to improve the sustainability and therefore providing more food for people.
ILRI and its international partners will continue to work together towards a greater understanding and application of the biologically diverse treasures of the world's plant genetic inheritance.